ECGs, and then we're going to set up the 12-lead ECG, which looks at the heart in 12 different vectors. It's technically 6 different planes, and we look back and forth in each of those planes. Actually, that's not even true. We'll see when we get there. We're going to set that up later today. But the lead 2 ECG with that stereotypical trace is one lead, one line, one imaginary line. And we only get a limited amount of information from that. The 12-lead ECG gives us a lot of information around the heart. Vertically, we can tell a lot of stuff about how the heart's working. If there's been damage to tissue, we can diagnose arrhythmias, tons of stuff. And we're going to get into that in the next section. But basic ECG setup. The rationale for an ECG setup is what we're talking about in this section. But first, I thought it might be nice to do some spirometry review. Just briefly, since you're thinking about this heavily in writing up your lab reports for this week. Spent a lot of time talking about COPD in that it is a chronic disease that's incurable. If it progresses, it can't be reversed. And what progresses are the chronic bronchitis, the chronic narrowing of airways, and emphysema, degrading the elastic tissue. No surprises here, but fairly important point. Those are the 2 constituent underlying disorders of COPD. The result is our ability to effectively transfer gas to the lung is compromised. So often these patients with severe COPD will display hypoxic blood gas, or they'll be hypercapnic, so high CO2 in the blood, and they try to ventilate more to alleviate those symptoms and increase what's called the work of breathing. So it gets really hard at the labor for them to breathe and move around normally because this gas exchange is compromised. And it's progressively more and more compromised as the disease accelerates. But we can evaluate how compromised a person might be. The forced vital capacity test you now have a lot of experience with. Full, complete inhale, massive, extreme, robust exhale, completely emptying the lungs. We get a sense not only of the static capacities, but the ability to move the air, which is the functional aspect of the test. In severe cases, most of these elements are affected, but in a strict classical sense, the obstructive types of diseases, where the airways are narrowed but volume's not affected, will affect flow rate. The capacity to load air and move air wouldn't so much be affected by just an obstruction. The air would move more slowly. But in the cases where lung volume is also reduced with pulmonary fibrosis, or if there's been surgery, or whatever other reason that might reduce the volume of the lungs, the restrictive diseases will show a reduce in the vital capacity, the volume of air that can move, whereas the airflow might not be affected. Combination of the two, which might be indicative of severe COPD, both flow and volume would be affected. Those characteristic elements that we paid attention to, one of the primary ones was FEV1, the amount of air you can expel in 1 second. This is a flow-related measure, how quickly air moves. It's often reduced in COPD because obstructions reduce flow. Without obstructions, that flow is not necessarily compromised. The restrictive diseases like fibrosis wouldn't affect FEV1. That's not to say that they wouldn't affect other measures, FEV%. FEV1 as a percentage of your total forced vital capacity. Well, that would be reduced in COPD, and it would be affected by fibrosis. Forced vital capacity is affected by fibrosis. Peak expiratory flow might be affected. The fraction of expiratory flow, measuring the middle of that curve, whether that curve collapses, indicating a gradual progressive obstruction, would change. We can monitor all those things and get a sense of, okay, well, is there an impairment and where might that be? Large airways, Small airways, how progressed or severe is it? Lots of information from just blowing forcefully through a tube, which is pretty cool. But none of this is new. You're all pros with this information. I just thought I'd line it up for you. Let's turn our attention instead to some new information. You are all probably in the camp of those that went through Bio 251, 252. So that was your anatomy and physiology 2 years ago-ish. And you probably remember all of the lead 2 ECGs, the membrane potentials, action potentials, graded potentials, EPSPs and IPSPs, excitatory postsynaptic potentials, inhibitory postsynaptic potentials. Anyways, we're going to review the stuff that's important for understanding the 12-lead ECG. There's a lot of terms that I just threw out that I don't expect you to remember. If you don't remember them, that's fine, because we're reviewing the pertinent information so that we can observe externally depolarization of the heart, the cardiac cycle, the electrical activity in the heart. I'll set up a 12-lead ECG for you at the end of this section, so we'll look at what how the electrodes are placed, where they're placed, what we mean by a unipolar and a bipolar electrode. We use both of them in a 12-lead ECG. The typical lead 2 ECG that you're familiar with is a bipolar electrode. There are some other ones that we use in a 12-lead ECG because it's difficult to place them all around the body. And then next week, we're spending time discussing how we can evaluate all of these characteristics. The 12-lead ECG gives us a lot of insight, not only into rate and rhythm, which is pretty typical. You can do that with a lead 2 ECG. But you can tell if there is a deviation in the net axis of the heart, if the heart has been enlarged, if there's an infarction or damage to tissue in the walls of the heart or somewhere in the heart. So we can tell a lot about the functioning and previous conditions or diseases that the heart has undergone using the 12-lead ECG specifically. And so we'll do a lot of examples and we'll go through these step by step next week. There are entire classes, like a semester's worth, 12 weeks of understanding the 12-lead ECG trace. So we are doing an abbreviated version. And we've done this for a few years, and I know a few students that have gone on to med school where they take up the 12-lead ECG trace, and they're just miles ahead, which is fantastic. Most students going in haven't seen a 12-lead ECG trace, so you get to run them in lab, you get your own printout, and you'll know how to interpret the 12-lead ECG trace, at least in a rudimentary manner, in a basic sense. So exciting stuff. Where did this all begin? The idea of animal electricity is at the foundation of our ability to observe depolarization in the heart. And this has roots back in the late 1700s, turn of the century, the 18th century, I suppose it's the 19th century, with Luigi Galvani that made frogs' legs dance by applying an electric current. This is at the time of the Renaissance, where people were chemists and biologists. He was a physician, a physicist, a philosopher, and he just studied how electricity affected dead animals in his spare time. The romance of the scientific method is kind of lost on us now. We study dehydration or lactate efflux, things like that no one really cares about. Not so much making frogs' legs dance, which is kind of neat. But imagine his surprise at a time where superstition is rampant to be able to apply an electric current to these muscles and have them twitch, have them move seemingly involuntarily. So Galvani was the father of, I guess we could say, electrophysiology. The concept that he came up with was animal electricity that has also been affectionately referred to as galvanism. And I'll show you, there's a video coming up of a galvanized corpse. The idea that you can reanimate dead objects, dead frogs, dead people. And it was also the root of Frankenstein as a concept. So this has some far-reaching impacts, at least in classical culture. The ability to make frog's legs dance doesn't really affect us at all. And it was in the mid-1800s that two physicians would take this work and use it to apply and make the same kind of thing happen in the heart. So just gradual experimentation, looking to see what tissues are excitable and how that excitability might confer differences in function. So at this stage, everyone is just poking and prodding and see what happens. They don't really have a firm understanding of why an electrical current might make muscle twitch. So Galvani has the idea that animals, people, frogs, mammals, reptiles, etc., have this inherent electricity that keeps them alive. And when you die, that electricity is gone. So if you could restore the electricity, certainly you could bring a person back to life. And this is an excerpt from a Discovery Channel video that dramatizes just that. This was an actual experiment. And this will be flagged for copyright, so it's not going to be in the YouTube videos, just FYI. So this is Galvani's nephew. Albini is Galvani's nephew. So he took up the work of his uncle, tried to reanimate a corpse and instill or infuse that electricity. The corpse was a convicted criminal that had been executed named George Foster. And you saw in the video, the fellow that procured the body actually died. This was at the turn of the 1800s. And this was an excerpt from the paper that reported the experiment on the first application of the process to the face. The jaws of the deceased criminal began to quiver. The adjoining muscles were contorted. One eye opened. In the subsequent part of the process, the right hand lifted and clenched, and the legs and thighs were set into motion. And so this spawned the idea of a galvanized corpse coming back from the dead. And you see the inset cartoon here highlighting a galvanized corpse. And the superstition running rampant back in the day thought that this was a medium for demons from hell to possess the dead and then try to entice the living and trick them into traveling down to hell. The caption, you can't read it here, but one of the captions says, "The thing begins to draw me. I feel it plainly, and now it winks. Oh, I'm lost. I can't withstand it. I shall hug it. Hug it. Yes, hug it." And then the guy falls on top of a corpse. So they're expecting that adding this electricity will trick people and cause them to go down to hell because they can't possibly abstain or hold off from the charms of this newly animated corpse. So, not quite scientific. This is more exploratory, certainly a little bit of fun if you're not the guy that procured the body. But our understanding of electricity has come a long way since then. We don't just poke and prod and electrocute corpses and make them quiver. We now have an understanding that there are specific potentials in the body, namely the resting membrane potential, which governs the ability to send signals to various organs, tissues, to control the intensity and the type of contraction. The resting membrane potential is central to all of the excitability of nervous and muscle tissues. And so we've since understood that it's set up specifically using ions of varying charges and then partitioning those ions within the body. And if we partition them to a certain extent, we can create a differential in charge. So yes, there is electricity, but it's not in the sense of a battery with flow where you hook up a current and you make electrons flow. There's electricity in a difference of charge or a differential across a membrane. And then we can manipulate that charge. And if we manipulate it in a number of adjoining cells, we can create what appears to be the flow of electric current through the body. So yes, electricity insofar as the cells exhibit a charge across the membrane. So we have a schematic, a very simple schematic of what we call a cardiac muscle cell, heart muscle cell. Any cell that has a membrane potential might exhibit the same kind of effects here. With the ions in question, the differential ions being partitioned being sodium and potassium. And you might remember from Bio 251, 252, we have a lot of sodium outside, very little sodium inside. But the counterpart is we have a lot of potassium inside, very little potassium outside. And by doing that, by actively separating those two ions, we can create a charge across this membrane that makes the outside more differently charged than the inside. We're not going to say how it's charged yet, but differently charged. We can measure an electric voltage across that membrane. So we have high sodium concentration outside, high potassium concentration inside. And part of the resting membrane potential doesn't only lie on where the charges are placed, but the ability to change the charge relies on how much of those ions there are. The concentration or the ability to move ions down their concentration gradients. The stimulus or the, I'm missing a word, but like the intention to move or the desire for the ions to move. They want to move according to charge and then down their gradient. So these wedges are meant to represent high sodium outside, and we want to roll down the concentration gradient in. There's that desire for these ions to move, and conversely so for potassium. So we have the concentration gradient, and the differential in charge is established mainly through one really classic ion pump, the sodium-potassium ATPase, which moves out 3 sodiums for every 2 potassiums. If you do that enough, you have more positively charged things outside than you do inside. And so the charge outside is more positive than the charge inside, which is more negative. And overall, if we measured the charge across the membrane, it's somewhere in the order of minus 90 or negative 90 millivolts, which is a fairly large membrane potential in the body. It can be as low as minus 120 or so. It can be higher depending on what cell you're in, but in a cardiac muscle cell, let's say a typical resting membrane potential is negative 90 millivolts. That means charge outside is higher than inside. Potassium wants to move out not only because the concentration gradient dictates as much, but also, sorry, I'm backwards. Potassium wants to move out down its concentration gradient. Sodium wants to move in down its concentration gradient, but also because the charge is drawing it inwards. We have lots of positive charge outside, which is repelling these ions, much like similar poles of a magnet, pushing it in into the cell. So this is the stage that is set that allows us to control or create excitability or an electric current in the body. We are going to look at this in a bit of detail. What I want to introduce to you first are the channels that are responsible for the movement of ions. This is a semi-permeable membrane. It can't allow the movement of ions by themselves, but channels exist within the membrane that allow ions to move. And some important ones, as we go through this in detail, are highlighted here. First and foremost are potassium channels. Potassium channels are voltage gated or concentration gated. They depend on a number of factors, but generally they allow passive movement of potassium out of the cell. They're passive movement of potassium out of the cell, which, if we understand the last figure, should happen. The concentration gradient wants potassium ions to move out. The charge gradient does not want potassium ions to move out, so those two are in opposition. Whether they move or not depends on the magnitude of those two factors. But if potassium moves out, you're removing positive charge from inside the cell. You're making the potential more negative. These are one kind of potassium channel. When potassium moves out, the potential becomes more negative. And I'll show you that on the next slide. One type of channel that results in the characteristic shape of depolarization are the fast sodium channels. These open really quickly, allow massive amounts of sodium to move through. How does sodium want to move? There's tons of it outside. So it wants to move into the cell down its concentration gradient. It also wants to follow the electrical gradient, positive charge outside to negative charge inside. So they move quickly, especially through these fast sodium channels. These allow the spike when we see a cell depolarize and the membrane potential to change a lot. We'll talk about these elements when we get to the diagram later. Lastly, calcium channels are not one that we've often discussed. Maybe you've heard them mentioned before, but they weren't on the last diagram. We don't really know what the concentration or charge gradient is for calcium channels or the calcium ion, but calcium is ultimately important in causing contraction. That's the early warning signal. In a heart muscle cell or skeletal muscle cell, calcium makes the apparatus, the muscle, contract. If it's there, the muscle will generate tension and shorten. So we really want to keep tabs on calcium. We want it not to be around if we don't want the muscle to contract. So we sequester it. We keep it locked within the sarcoplasmic reticulum. But we do manipulate it specifically when contraction is desired. So we have these slow calcium channels that allow calcium to move out and contraction to happen. Being that they're slow, it allows a sustained contraction. The slow nature of these channels is one of the things that differentiates cardiac from skeletal muscle. Skeletal muscle is generally faster, and hopefully show you how that comes into play coming up. So in very general senses, these are the three main ingredients in the cardiac cycle. What do they look like if we trace or we follow membrane potential through an entire cycle? So we said that resting potential is somewhere down here. We're going to say it's negative 90 millivolts. And then something happens that causes a massive, dramatic change, a rapid increase in that membrane potential. And you know this to be the rushing in of sodium. Voltage-gated sodium channels open. Sodium rushes in. More positive things inside means the membrane potential spikes, it skyrockets. This is one characteristic that initiates the cardiac cycle, or initiates depolarization. Initiates contraction. This shows you depolarization. Depolarization is membrane potential going up. As it goes up, it is depolarized. Down here, it is polarized. There are two very different charges. As it increases, they become more equal. At 0, the cell is fully depolarized. When we have this spike, there's a prolonged plateau in a cardiac muscle cell. And this prolonged plateau is important for the squeezing, the prolonged contraction of cardiac muscle cells. It has this shape because of those slow calcium channels. Calcium is persistently released during this phase. Persistently released. And it's balanced by the movement of some potassium so that there's no net change in the potential, but we get this infusion of calcium. This sustained squeezing and contraction of the cardiac muscle cells, which is important for organizing the flow of electricity through the heart. We'll come back to that. We have to restore everything to normal. We can't initiate a new heartbeat or a new cardiac cycle from here, so we repolarize the cell. We reintroduce that resting membrane potential by getting rid of a lot of those positive ions that have now flowed into the cell. So we move out potassium, we move out calcium, we move out sodium, and gradually removal of many of those positively charged ions moves the membrane potential back towards our resting negative 90 millivolts. These points are all vague. They're not detailed, but this is probably in line with your understanding of the cardiac cycle or the depolarization of a cell coming into the class. Something along those lines. Channels open and close, ions move, membrane potential moves accordingly. And this corresponds with phases of the cardiac cycle that we use to make sure that everything happens in order. We know that depolarization occurs as the membrane potential rises. When a cell is depolarized, it doesn't have radically different charges inside versus outside. So the blue area is the depolarized phase. Contraction occurs once we see the release of calcium into the cell. Notice they almost overlap perfectly. The signal to release calcium is at the start of the blue bar. The release of calcium is at the start of the orange bar. So there's a small amount of time between sending the signal and then having the muscle contract. Contraction lasts almost the entire period, and importantly, this is governed by an intrinsic refractory period. And what that simply means is that a cell that's been depolarized can't be repolarized or can't trigger again for a certain amount of time. We can't have multiple depolarizations happening in quick succession. We wait until the end of the refractory period before the situation is returned to normal to be able to be excited again. Cells are immune to stimuli during this refractory period. What's really important about this is that it makes sure there is an ordered hierarchy. If you have one cell up here that's depolarizing and sending signals distal within the heart, you don't want that signal to move around or back up. You want it to move in one direction only. And the refractory period makes sure that anything that's been depolarized initially can't be re-stimulated to ensure that the signal continues in one simple direction. Let's put a little bit of detail to these points. And this is overly complicated. We won't talk about all of these elements, but I want to give you a bit of an understanding over what specific ions move and when. And so this figure is set up with a very similar scale to what we just saw. Resting millivolt potential on the y-axis. The x-axis is time. You can see we're at about the resting membrane potential that we identified earlier. The red line is what happens to the cell during the cardiac cycle. This is, again, a cardiac muscle cell, so it's a very similar setup. The currents are indicated by arrows, so all of these boxes represent specific ion channels. The I means flow. The subscript means what ion is flowing. And there might be a little addendum to say, okay, potassium is flowing because of the actions of acetylcholine. This is an acetylcholine-gated potassium channel. We don't care so much about that, but just if you're looking at the subscripts and you're confused, there's information about what triggers the channel. So each box is a channel, and the arrow indicates where that ion is moving. In this yellow box, sodium is moving into the cell. In this box, potassium is moving out of the cell. This NCX box down here is not as obvious. This is sodium-calcium exchange with a net flow of ions in. A net flow of sodium in. So you can decipher what boxes or what ion channels are open at any given point in time with these boxes. You can also break it down by color. Yellow is in, gray is out, if that helps. One other important thing to note before we go through this in detail, there's a small aberration at the end of the figure, this dashed line. We're not paying attention to that right now, but this represents what happens in a special kind of cell, the pacemaker cell in the heart that dictates when the cardiac cycle fires. So a normal muscle cell follows the solid red line. A specialized pacemaker cell follows the dashed red line. And I'll show you what that looks like in a full trace when we get through our discussion of this diagram. These dashes along the bottom also show you the periods of time for which each of these channels is open. So this potassium channel is open throughout this horizontal solid bar at the resting phase. So let's talk about the resting phase. Resting is referred to as phase 4. Anything with a normal resting membrane potential is considered the resting phase. In this phase, a cell is able to be excited. It's able to be stimulated. It's able to depolarize. It's ready to receive that signal. Generally, membrane potential is unchanging. It's pretty flat in a normal cardiac cell. Some channels are open. So on the left-hand side, these boxes indicate that potassium is leaking out slowly. Potassium is leaking out slowly. And it will leak out, according to the concentration gradient, inside versus outside the cell. And in fact, if you have high blood potassium, you can really disrupt the outflow of potassium in these cardiac muscle cells and induce cardiac arrhythmias. The outflow of potassium is very susceptible to blood potassium levels. It's typically very low, and if you over-supplement or have a hyperkalemia for whatever reason, high blood potassium, you can really disrupt this resting phase. There is gradual leak until something happens. The thing that happens is either the cell receives a signal, and that's what's going to happen in our example here, or if this line wanders too positive, it will trigger a depolarization. But being a normal cardiac muscle cell, this cell will receive a signal and respond accordingly. What is the response? The response is characterized, don't worry about that. The response is characterized by phase 0. Phase 0 is called the upstroke. Phase 0 is the spike in voltage. When a signal is received by the cell, these sodium channels allow sodium to rush in. There's a specific channel that responds to a change in voltage. When the voltage reaches a certain level, they open. When they're open, sodium runs down its concentration gradient. It runs down the electrochemical gradient, rushing into the cell, making the inside of the cell more positive. And so we see that trace here as this line spikes. It's almost completely vertical, a near instantaneous influx of sodium. Now you might remember this signal being sent via nerves, right? If a nerve depolarizes a muscle, that signal results in sodium rushing in. The other thing that happens, did you learn about gap junctions in 251, 252? So any cells that are connected to this cardiac muscle cell allow the flow of ions through gap junctions, holes between the cells. And so we have natural flow of these ions between that changes the membrane potential. When it reaches a trigger point, sodium rushes in. Neither of those cases, membrane potential reaches a threshold, rapid influx of sodium. Now this is the signal. This is what says cell, contract. Contraction doesn't happen immediately. What we need to do first and foremost is protect the status quo. We need to make sure that we maintain membrane potential and we don't lose the potential across the membrane. If we do, then there's no incentive for ions to flow. And so the way that we do that is we catch the membrane potential as it spikes. This is often referred to as the latch or the notch on the cardiac cycle diagram. If we simply let sodium rush in, you can imagine a theoretical point where all the sodium that possibly wants to rushes into the cell. And at some point, there will be enough sodium that the amounts inside and outside are balanced. And if they're balanced, there's no incentive to flow in and out. We lose the membrane potential. We don't want that to happen. So, the heart is designed to stop the membrane potential from going too high by quickly opening and closing, by spiking open these specialized potassium channels. What this allows us to do is catch the membrane potential before it gets too high. Think about it. Sodium's rushing into the cell. Everything around me, if I'm in the cell, is getting more and more positive, more and more crowded with sodium ions. I still have a lot of potassium as well. If I open up these latch channels, it's like a blow-off valve. It relieves the pressure. There's lots of positive pressure, lots of ions in the cell. As these open, okay, potassium can rush out quickly. Potassium's positive. Potassium moving out means there's less positive stuff inside. Membrane potential drops sharply, and we maintain some sense of an electrical gradient. If we were to reach a sodium equilibrium, we would have a really hard time restoring that division that's characteristic of our resting membrane potential. So that's why these potassium channels snap open and shut really quickly as a blow-off to relieve some of the pressure. Now, we are in the optimal scenario for contraction to occur. When we reach this relatively stable, somewhat positive membrane potential, that's a signal for calcium to be released. Calcium is released from the sarcoplasmic reticulum as soon as it's released, as soon as it's in the cell, it binds to troponin and initiates contraction. Any amount of calcium that's present in the cell will do this. Small fractional amounts of calcium will stimulate contraction. We keep really tight tabs on it within the cell. And so as soon as calcium is released, the muscle starts to shorten. It generates tension, starts to squeeze. Calcium is released in this ideal, somewhat positive scenario of membrane potential. Since we are releasing calcium, this is doubly positive. We also have incentive for the inside of the cell to become more positive. We balance that incentive by allowing some potassium to move out. Different cell, or different channel than the latch channels that open and shut really quickly. Since we have this influx of calcium, we balance that with some outflux of potassium. The main effect is that this plateau, this flat area, flat-ish area, persists and allows contraction to occur. If we didn't also allow potassium out, this would start to wander up, which is something we don't want to do. That's why we had this latch mechanism earlier. So we are in a scenario with some potassium in, but a lot of it is flown out. Lots of sodium in, because it's rushed in during depolarization. Lots of calcium in, because it's been released to induce contraction. That's not the resting state. It's wildly different from the resting state. So in order to restore the resting state, we need to do a few things. Here, this is what's happening currently. Really high sodium inside, high calcium inside, low potassium inside. We don't want that to occur. We want no calcium, and we want the sodium and potassium to switch places. We want the opposite, right? We want high potassium inside and low sodium. So how do we do that? First, we shut off the supply of calcium. We don't want any more to infuse. Contraction is done. Now we need to tightly regulate calcium again and store it away. Shut off calcium channels so there's no more influx. We open potassium channels just to bring the membrane potential back down. This is the only ion that we can allow to flow naturally and restore membrane potential in the direction it's supposed to go. Potassium will flow out on its own. Removing positive charged ions from inside the cell makes the membrane potential go down. If we opened up sodium channels and let more in, membrane potential would go up. We don't want that. If we let more calcium in, membrane potential would go up. We don't want that. So we open potassium channels that allow potassium to run out to initiate this drop in membrane potential, this final repolarization. Which is good because it brings us back towards our resting membrane potential, but it's bad because it It's bad because, let's just take this off. There. It's bad because the potassium is outside. We want it in. So that's when we need to be active in restoring our normal balance. And these are the main players in restoring the normal scenario. Sodium outside, potassium inside, calcium put away. During this repolarization phase, all of these pumps or channels are open and active. Sodium-potassium ATPase uses energy to move sodium out and potassium back in. It's going back towards normal. The calcium ATPase, also known as SERCA, sarcoplasmic reticulum calcium ATPase, pumps calcium back into the sarcoplasmic reticulum. It puts it away. We don't want it around in the cell. If it's there, it'll start a contraction again. We want it put away. We want tabs on calcium. And the sodium-calcium exchanger, which we mentioned briefly at the start, again, will relieve any calcium in the cell. It pumps it outside of the cell and exchanges it for sodium. I didn't put the numbers up here. There are, hold on, am I going to get the right number? Yeah, 3 sodium in for every 2 calcium out. So more sodium moves in than calcium moves out. This is not what we want to maintain normal potential, so the sodium-potassium ATPase is working extra hard to make sure we restore that gradient. The net effect is we're back at a resting potential. Calcium's been put away. It's either outside of the cell or stored. If we have a differential in sodium, we pump that out in exchange for potassium. The result is calcium's put away, sodium's out of the cell, potassium's in the cell, we are back at rest. All of that long-winded description happens in a fraction of a second with every single heartbeat. So you're sitting here, and in that amount of time, you probably had 200 plus heartbeats, maybe more. This happens between every beat. One beat happens as we depolarize the cell, and then it returns to normal before the next one. All of those ion channels opening and closing, the flow of ions in, refractory period, moving ions out to restore normal balance, all that happens in fractions of a second. 0.2 seconds is shown here on the graph to give you a sense of the scale of time for all of these ions moving in and out. So rapid rush of sodium in, we catch membrane potential with potassium flowing out. Calcium persists and makes this plateau, contraction occurs, and then we allow potassium to flow out to bring membrane potential back down while constantly pumping to restore our normal scenario. A brief overview of what happens in our cardiac muscle cell. So this is what the trace would look like for a cell that responds to a signal. What makes the signal? The pacemaker cells are those that make a signal, and a general trace for the waveform of a pacemaker cell is shown here. Notice the characteristics of this graph look fairly different. First, these aren't nearly as depolarized. Resting potential in a pacemaker cell is higher. Instead of negative 90 millivolts, we're sitting at negative 60. So it's easier for these to trigger. Good. Well, we want the pacemaker cells to be the ones to trigger. So if it's easier, that's fantastic. These also are much leakier. Much leakier. What that means is there's more sodium flowing in, and this gradual rise in membrane potential occurs more quickly than in a cardiac muscle cell where it's clamped, where there isn't easy movement. What does that allow the pacemaker cell to do? As sodium leaks in and membrane potential creeps up, we reach this theoretical point identified here by this horizontal dashed line. This is the trigger potential. If membrane potential reaches this point, the voltage-gated sodium channels open, depolarization occurs. Pacemaker cells will always eventually run into that point and trigger a depolarization. They do that quickly and easily because there's a sodium leak into the cell. We want that to happen quickly so that it initiates the signal and other cells in the heart don't. Also note, there's no prolonged plateau. This entire cycle is not drawn out. There's no contraction happening in these pacemaker cells. Their sole job is to create the signal and send that signal to muscle cells that will contract. So we don't need the plateau because we don't need contraction. That has the added benefit of making the entire cycle faster. We don't need a long, drawn-out plateau. We need quick depolarization, repolarization, depolarization, repolarization. The entire cycle is compressed. So these things will trigger more quickly, trigger more frequently, and then send signals through the pathways in the heart to make sure other areas also depolarize in time. No other cell should trigger before they get a signal from the pacemaker cells.